Good morning, everyone, and welcome back to day 4 of the workshop on finding cancer drug targets to facilitate precision and personalized medicine. We look forward to another engaging and productive day of learning together. Today's program begins with a lecture by our host faculty, Dr. Mohammad Azhar Aziz, on identification of novel therapeutic targets in colorectal cancer.
All right. Thank you, Swati. Good morning, everyone. And Assalamu alaikum. So today we are going to go further from what we have learned so far and dive into the actual uh, problem of finding the therapeutic targets. So I think we have already covered what is multiomics. So I hope all the participants here would be having now the concept of multiomics clear, what it is. Then I also covered how do we do it. And yesterday tutorial showed you some computational tools by which you can do multiomics. You can test your gene, you can test different databases, DepMap, UCSC Xeno browser, the Protein Atlas, and you also have a wealth of data in the TCGA, the Cancer Genome Atlas. And then we have an AACR also maintains a database. So there are a lot of data that is available out there, but we need to work on it and make out sense so that we can identify novel therapeutic targets. Now, remember here, we are not looking for something very different, but we are trying to find out the targets that are for a particular patient. So that is the motive. It's not about finding new gene every time, new mutation every time. Rather, it's for finding the right gene for the right person or right target for the right person. Okay, so I will move my slides by setting the stage with the problem of colorectal cancer to be specific. So what you see on your screen is about the incidence in blue and mortality in red from all over the world. This is a slide for the global numbers. And you can see that there are roughly 1.3 million cases in 2022. From all ages, from all regions, 1.3 million people that are diagnosed with colorectal cancer. And roughly 40% of them, they die. Right? So this is still a problem, although this is a better statistics, because now due to preventive strategies, surveillance, and screening procedures, some countries like United States and New Zealand, they have reported a decline in the mortality. But when you take it globally, it's rising up in India, it's rising up in other countries of Africa. So this is still a major global health problem. Let's talk about India. So we have to have something specific. As I told you in my earlier slides, we can't generalize it all over the world. Whatever works in other countries may not work for us and vice versa. So I was looking into the numbers for India. Here also, although the numbers are low, but the survival rate is pretty bad. It's just 50%. 50% of people who are diagnosed, they die. So this is all stages, all age groups, and all genders as well. So the magnitude of the problem is quite big, and this has been. I'll show you the problem that is coming up with the survival rates. So far, we have got these strategies for treating cancer: the primary surgery, then chemotherapy, the drugs that we have talked about, which are working in a systemic manner that goes in the entire body and try to target the replicating cells, the dividing cells, and kill them so that the tumor is also eliminated accordingly. Then radiation therapy is used in some cases to shrink the tumor. Sometimes the tumor is pretty big, and there is a need to shrink the tumor, and for that, radiation therapy may be used. When the options of systemic therapy, chemotherapy are not working, and the stages are advanced, then there is targeted therapy comes into play. I don't understand why they don't use it to begin with. Probably it's about the cost. It's about I don't know the reasons why they don't start with targeted therapy, but this is what it is. The targeted therapy is given to those patients who have advanced and who have not responded to chemotherapy. And the latest on the block is immunotherapy, that there are ways to boost the body's immune system, and by using the checkpoint inhibitors like CTLA or PD-1, PD-L1 inhibitors that can be utilized for curing. But this is also in the advanced stages. So we have got all these modalities available for treating the colorectal cancer patients. But still, if you look at the survival rate, so I'll walk you through the slide. These are the stages, which is called as Duke's staging system in colorectal cancer. It's in the alpha numeric format: A, B, B1, C, B1, B2, C, and D. Then we have got the tumor, nodes involved, or metastasis. This is called TNM classification. So if there's a tumor, which will be there, and if there are nodes, which are lymph nodes, are found swollen in the body, we call it as N. And if there are metastases, which means it's already spread, then we mark it as N. So if you see here, T1 means there is a tumor, but there's no node involvement. There is no involvement of there's no metastasis. So this is termed as stage 1. And pathologically, I'll show you in the next slide, what does this mean? That cancer is limited to the mucosa, which is on the intestine, which is on the surface, and survival rate is pretty high. Greater than 90% people, they live, and the surgery is the main option here. Once the tumor grows in size, it's T2, but still there's no node involvement, no metastasis, but the cancer has now started going into the layers of the muscle. The chances of survival is dropping. As you can see, the chances of survival is dropping, and it is drastically low in case of the advanced final stages of stage 4. And you can see here the tumor size is growing, and then you have got the node involvement, and then you have got finally the metastasis as there as well, which is stage 4. Right. So these are the survival rates, which again remind us of the magnitude of the problem, that we need to take care of these patients, despite of having all these treatment modalities available. So this is another slide showing the same numbers in a more graphical way. So here is the start of the colorectal cancer, which is benign so far, which is on the surface. This is the surface, and this is the building of the tumor, the hyperproliferation. Then we have got the adenomatous polyps. Then there's a precancerous polyps, and then it's a full-blown adenocarcinoma. But once it starts going into the muscle, the underlying muscle, this is where the problem is severe, because the next step is the metastasis. And this is the region where it's malignant, means cancerous. Benign means it may stay there forever without doing any problem. But malignant means it is now going to spread. And then these are the survival rates, as you just saw in the previous slide. So as we advance into the cancer stage, the chances of survival are low. This tells us about two things. Number one, the targeted therapy may be started early on. As I said, I don't know how these clinical decisions are being done. Is it driven by the cost of the drugs or whatever? And second thing is, if we are able to detect this disease early on, then the chances are very, very good. So we can develop both the screening and preventive models and preventive strategies by the help of the biomarkers. And we can also work on therapeutic targets that can help us in developing early therapeutic targets for early intervention. Now, let us see what are already known to us. And still, the statistics is so poor that people are still dying. So this is the pathway that is very much related to the colorectal cancer, the EGFR pathway, which means there is a cell surface receptors, epidermal growth factor receptor, and this is driving the rapid proliferation of the cells. So if we are able to block it, usually this is the ligand, epidermal growth factor, which if binds to this receptor, the cell starts to grow. So if somehow we are able to block this, we hope that the cells will stop to grow and the tumor will be cured. For that, there are two antibodies, cetuximab and panitumumab. Both are having the same target, but one of them is humanized, which means it has got one chain. Antibodies have got two chains, right? If you remember, the the Y-shaped structure. So the one arm is there from the humanized, and the other one is the mouse one. Cetuximab is entirely derived from the mouse. And then there are downstream pathways. So we already have these drugs which are given, but still there is not much improvement. Then we have got another pathway, which is vascular endothelial growth factor pathway. So this helps in the building of new blood vessels. So there are three types of receptors here: VEGFR receptor 1, vascular endothelial growth factor 2, receptor 3. So they have developed antibodies to block these as well. So there are different drugs which are targeting different formats. Like this is aflibercept is going to disrupt all these three ligands. Bevacizumab is also going against VEGF-A. This is the ligand here. So they stopped them from binding so that this formation of the new blood vessels is stopped, because the presumption is once you stop the formation of the new blood vessels, you can starve the tumor to death. Then they will not get any new blood supply, they will not get nutrients, and then they can die. But and there's another one. This is and regorafenib is a drug which can be given to block all these three together, these three receptors together. A wonderful drug, a wonderful target, everything known. Still, the statistics is pretty bad. 5% survival. Remember that. Only 5% people are able to survive in this. Okay. So next is the target HER2, human epidermal growth factor receptor. This is also being targeted by cetuximab, panitumumab through EGFR, because EGFR HER1 interaction can be disrupted to prevent the cell from binding. Trastuzumab is specifically for HER2. Then in this intracellular domains, there are different drugs that are available for blocking this entire pathway. But cancer cells are really smart. They know how to work around, and even if you block this one, they might take this pathway, or they might take another pathway, and they can still survive. So this is what a normal signaling looks like. And here, when you block the signaling, we are presuming that we block the receptors, so everything is blocked, but it does not happen in the actual scenario. Another target was NTRK gene fusions. Yesterday, Dr. Asim was telling you about the gene fusions as the causative events in the DNA that is leading to cancer. So like CBR, ABL mutations in the blood cancers in the kids. And in case of colorectal cancer, we have got NTRK gene fusion. This is also there in the lung cancer. That gene fusion is like gene from NTRK gene fuses with the other gene. And this fusion, this coming together makes a lethal combination that helps the cells to grow indefinitely. In fact, it's a kind of mutation that helps in the rearrangement and the formation of these fusion genes. And it is common in colorectal cancers with microsatellite instability. I'm not going into the details of the types of colorectal cancers, so that there are different types of cancers within the colorectal. But what the message you have to get is that the targets were identified based on single omics, and then they are making things better, but not to the extent that we can have full control of it. Another example here about the microsatellite stable tumor and microsatellite unstable, or the tumors which are deficient in the DNA repair, which is mismatch repair here. If we have got the tumor which is having the mismatch, the microsatellite stable, then the tumor is not expressing a lot of new antigens. So remember here, we had a discussion that if you have got a faulty DNA repair system, is it good or bad? In this case, it is good. Because once you have got a faulty DNA repair system, the DNA is having errors, and this is leading to the production of more antigens which are new. So this is the difference that you can see here. This is a stable tumor. It has got only one epitope here, which is being recognized by the T cell, right? So the response is quite poor. But if you have got a microsatellite unstable one, more errors are being done in the DNA. The mutations are more there. So we have got more new antigens, which are being recognized by the T cell, and ultimately you see a good response. So now it is well established that you don't give this kind of a therapy of anti-PD-1 or PD-L1 antibody to the patients who are having good DNA repair system. So it is given only to those patients who have got a poor DNA repair system. A little bit of personalized medicine that you can see here. In fact, in this also, if there are KRAS mutant patients, so this is a RAS gene, if it is mutated, then it does not need all this dimerization or binding of the EGF. Rather, it works on its own, and it can drive the cancer to cells to grow more and more. So if there is a KRAS mutation in a patient, they are not given the therapy of this, so that because it's useless. It's not going to work. It's already mutated, and it's already going to drive the cancer cell growth, right? So it's very important to know the patient profile before we give them any kind of drugs. So we are stepping towards that, but we are not yet close. So the question that comes out is, we have seen so many targets here: the EGFR, the HER2, the PD-1, PD-L1, so NTRK fusions. Why these targets fail to improve the therapeutic outcome? I will give you a few seconds to read this and see what is the current scenario in India. I'm not talking about the whole world. I'm talking about the country where I'm speaking from. I think I have a question here. Chewy, I'll have to take time off to do this job, but you can ask your colleagues if somebody has it, or I'll ask the Dr. Afzal if you can provide the links here in the message box. Okay, so globally, India ranks 5th in the CRC mortality with 38,000 deaths in 2022 alone. And still, if you see the 5-year relative survival of colorectal cancer, it's pretty high in case of US and other countries. So India is just second to China in case of the rectal cancers, which is an alarming situation. But still, we don't see them in the priority list when they do the funding. They don't have any priority for colorectal cancer. So are we really seeing the right target, and are we really treating the patients with the proper knowledge of what works in this particular patient? If you can remember this elephant story, and this jigsaw puzzle. If we are not knowing the complete picture, then we are really missing something and we are missing the target. And this is applicable for different patients. There will be different targets. So remember, we have to put the picture together. And this is something that I'm going to tell you with some examples today that people have done and published by using the multi-omics approach. So how to put together the full picture? That's the question. So if you remember, we talked about multi-omics, that we need to put things together from the DNA level, from RNA level, from protein level, and metabolite, as well as many other omics to create the full picture. And then we can say that, yes, this is the right target for this right person. That's the precision and personalized medicine. So this is the latest report which came out in Discover Oncology. They identified a therapeutic target in lung adenocarcinoma using Mendelian randomization and multi-omics. So people are now acknowledging this, that yes, this is the right way to do it, but still we are not there. We are still have to sharpen our tools to find out how to put the jigsaw puzzle together, because it is very, very complex. It is not as easy as we usually see. So through the multiomics analysis, they found out ICAM5 as a primary pathogenic target in lung cancer, right? And they had a significant impact on how the disease was going to progress and how it was going to respond to the drugs. And additionally, they also found KLK13 and FUT8 genes. They can be the potential pathogenic target. This was, again, a population-based study, not a longitudinal study. So, but yes, they refined the population and they subtyped it so that they can have better handle on who is going to be getting benefited from this. Another report that came out in Cell Reports Medicine, a very nice paper, a multiomics analysis assisted deep learning model. So here you see the use of machine learning and artificial intelligence, and they were able to find out a macrophage-oriented module. They did not find one target. Rather, they found this module. This is the module, which means they were CD. If you are not familiar, these are all the immune cells. They were exhausted immune cells, CD8 plus T, CD4 plus T. Then there were T regulator cells and tolerant CD8 plus T cells, and the whole R2 plus macrophages. So this was the module that they were able to find out that was the right target. So they used the deep learning models in the macrophage-oriented spatial interaction map. So the study, if anybody is interested, they can look at that. This is also from last year. It's a pretty new one. And once they targeted the full R2 plus resident macrophages, that enhances the response to chemotherapy. And now look here, the chemotherapy was given, but it was given along with this full R2 plus targeting. If you can see the survival probability was pretty high in case of this combined treatment. So the chemotherapy response predictor was designed, and then depletion was done as well as chemotherapy, and tumor was found to be regressed. Regressed means it was the size was reduced. So this is another example of using multiomics, finding out a target, and then using it alone or along with the chemotherapy. So this was used along with the chemotherapy. And I'll tell you some story about our own work, what we did in this direction. And the story starts from the patients that we took the samples from. They were all colorectal cancer patients. We took their tumor and normal tissues. We extracted DNA and RNA and protein from them. And then we started to do the study step by step. So first block, we did the genomic study. And this is the microarrays. So that's what I was telling you yesterday, that we did the microarray-based study. And we were able to find out these changes So these are the cytobands, which means the portion of the chromosomes that were found to be affected. And these are the genes that were known to be involved in colorectal cancer. But these were the genes that we were able to find out as novel. And there's a score which tells us the confidence that yes, this event and this gene is uh, pretty robust. The possibility there's a algorithm which is a GISTIC. It was developed by a group in Harvard. Gatti Getz is the person who did it. So it's known as genomic identification of significant targets in cancer, GISTIC. So that assigns a score. So that score helps us to give confidence about the targets that we are identifying. So we found out 11 new genes here. And out of them, we were able to zero down upon a few of them and take them further. So total 144 genes were found as significant targets, and out of them, 11 were novel genes. Now, here the approach that we took was highly personalized. We analyzed each patient separately. It was patient level analysis, not that we mixed all the tumors patient together, cancer patients together, and all the normal samples together. No. We did patient-wise analysis, and that gave us this high-resolution picture and confident targets. Then we tried to put another level of omics, transcriptomics. So we took the same samples from which the RNA was extracted in the same protocol, and we ran the exon array. Exon array is meant to measure the RNA levels. When we say exon, because exon is part of RNA and we are seeing it at a higher resolution level, but the information that we're getting is about the RNA level expression of those genes. Now, this was the result using two different algorithms, alt analyze and expression console. So this was the pattern of the gene expression. So these are the gene expression. Now you will be astonished to see that whatever was changed at the DNA level, not all of it was changed at the RNA level. Only 20 out of the 144 genes were found to be differentially expressed. So that's why whatever is we are seeing at DNA is only part of the information. It's part of the picture. It does not necessarily be the same at the RNA level. So only 20 of these genes were found to be significantly altered at the RNA level, right? And from here, we picked up a few genes for further analysis and further work. And one of them was ADB8B1. And some of these genes were very interesting that if you look at this one, ABP1 gene, this was in the region of the genomic gain mostly, which means the copy number was more in the tumor. But if you see at the gene level, expression level, it was downregulated. So at the DNA level, the copy number was more, but at the RNA level, it was down. So it was a complete paradox. So we suggested that there would be some involvement of the transcription factors that is showing this phenomena. And if you read the paper, you'll be able to get the full picture of it. But there were three other genes which were showing the same scenario, like BCAS1. It was having 80% gain, which means the DNA was amplified. So usually you would expect that the DNA is amplified, more DNA means more RNA. But no, here it is downregulated. The more DNA, less RNA. So this is not a linear relationship. I think Dr. Asim also mentioned that. Similarly, from RNA to protein is not a linear relationship. This is a relationship which needs to be understood through mechanism. So we need to know both levels, genomics as well as transcriptomics, to create the full picture, right? So now I would ask you, can you figure out which genes are altered both at DNA and RNA level? And now, if you look at this, I'll go back to my slides. So look at this DNA level changes. Look at the genes within the red box. I'll give you a few seconds. If you want to take a screenshot, you can do that as well. And then look at this picture where you have got the RNA level evidence. So you have to spot out which gene or genes are common to both DNA and RNA. Anybody with the right answer can win a Swiggy coupon. I may say that. Nobody? We have got 53 people in the meeting. If you want, I can show you that again. Okay. It's a very quiet class. So if you look at this, ADP8B1 is the one that is significantly altered at the RNA level as well as at the DNA level. This is at the DNA level changes. Very good score, 2.326. And this is also down-regulated in case of the RNA level. So it's like the elephant story. If two people are saying the same thing, you would believe them more than one person saying it, right? So here we are having two layers of evidence saying the same thing, that ADP8B1 is probably a good target. DCAS1 is there as well. And there are other MUC4, ADAM12. I'll come to that story again. So the point here to convey is that we are trying to recreate the picture using two different blocks of the evidence at the DNA level and the RNA level, and then we are more confident about the target itself. So it's ADP8B1 and there are MUC4 and ADAM12. These are all the genes that were found to be altered at DNA as well as RNA level. So that's why I'll tell you the story about the ADP8B1, which was a very probable molecule, because we also know that this is the molecule that is found on the surface of the cell. The cell surface proteins are also very good drug targets. In the DNA and RNA, we might find the target, but we may not be able to drug it. We may not be able to do anything about it. Although now the CRISPR technologies and they are allowing us to do the gene editing, but still the proteins, and especially the proteins that are available on the surface of the cell, they are the favorite among the drug discovery people. This ADP8B1, we discovered it to be associated with colorectal cancer. And since it was downregulated, we proposed that this is a tumor suppressor gene. Let me show you what we did next. So this is the data from the patients that it's showing that it is downregulated. Then we saw it in different patients individually. Yes, it was all downregulated. Then we tested it in the cell lines. Now, cell lines are different. You can see only two of them are showing down, or three of them are showing downregulation. The rest of them are showing opposite, upregulation. So we needed a good model. So this was perhaps SW480 will be a good model, or GD116 will be a good model. These are all colorectal cancer cell lines. Right. Then we used the shRNA to knock down the ADP8B1 gene. Once we knock down the ADP8B1 gene, what will happen? It's a tumor suppressor gene. If you knock it down, what will happen? Sorry, Padmavati, we just moved. Can you answer this question? If we knock down the tumor suppressor gene, tumor will grow. Very good, very good. So tumor will grow, and you can see in this green line is showing better cell proliferation rates than the control. So this is showing that the tumor is growing faster and giving us a hint that yes, your gene is a tumor suppressor gene. Next, we utilize shRNA because we always use multiple evidence. Now we try to put ORF. ORF means we try to put another copy of the gene into the tumor cells. Now, what would we expect? If you overexpress a tumor suppressor gene, what would we expect? Anybody? Absolutely. Thank you, Padmavati. And Mustafa, it will reduce the tumor growth. And that's what we saw, that in this green line, the tumor cells were growing slower than the control one. This is the proliferation rate. And this is a very nice equipment. It's called Excelligence, which gives you the real-time data about the growth of the cells. But this is all inside the cells. We need to test it in the preclinical models, and I'll tell you about some preclinical models as well later on. So gives us in fact, we also use CRISPR. I have not shown you that data, but CRISPR and shRNA, they were able to increase the proliferation rate, whereas the ORF, which is open reading frame, which means we are putting another copy of the gene, which was able to suppress the growth of the cells. So this proved to some extent that this ADP8B1 gene is a tumor suppressor gene. But how is it working? It's still unknown. And if anybody wants to take up that as a molecular biology problem, that will be wonderful. Because ADP8B1 is a molecule on the surface, and the mechanism by which it is working as a tumor suppressor is still unknown. Now, we have got another piece of the puzzle, which was metabolomics. So we did not have any setup for doing the metabolomics at that time. So we collaborated. This is where the tutorial about consortium and other things would be helpful for you, that you need to collaborate. You need to go across the department and shake hands. So I contacted a person in Sweden, and he gave us the genome scale metabolic models. These are known as GEMS. So we take our transcriptomics data, and he fed it into the genome scale metabolic models and identified some targets. And we also did the PCR validation, quantitative RT-PCR validation, to find out which metabolic pathways were significantly altered in our patient samples. So this is a complex pathway, but I can tell you that this was the glutathione metabolism, which was getting affected in these patient samples. And the genes that were affecting that was ODC1, SRM, and SMS, SMOX2, and RRM2. So we collaborated, and we found that probably these are the target genes. Now, these genes were differentially altered in different patients. So again, personalized medicine would be helpful. We look for these genes in individual patients and see which one of them can be helped by targeting these genes and which one of them will not be. So here I will introduce for some early learners this technique that we used for doing the validation. I missed that yesterday. So quantitative real-time PCR, which is going to give you an idea about the copy number of mRNAs present in the sample. So we can use different chemistries. There is Stackman probe, there's molecular beacon, and there is a simple one called cybergreen. It's not here. The cybergreen is a dye that just binds to the double-stranded DNA, whereas Stackman probe and molecular beacon are very specific for your DNA. And when they bind, they also emit signals, light, which is recorded. So as you do the PCR, I'm presuming that all of you know about the classical PCR. With every cycle, you are amplifying the number of the copies of the gene. As you are amplifying, increasing the cycle number, the fluorescence intensity also increases, and it gives you an idea about the amount of RNA that was present to begin with. So if you've got less RNA to begin with, it will take more cycles to come to the threshold. If you've got more material to begin with, it will take less cycles to come up to the threshold level. So we utilize this technique, and we validated these genes. Now, if you look at, these are different patients. And in these patients, they are upregulated, while in some patients, this is downregulated. So if I'm to give a therapy which is against ODC1, in fact, there was a drug that was in clinical trial, tazarotene. It worked in some patients, did not work in some patients, and it was stopped because half of them were not treated. So here you can again appreciate the personalized medicine approach, that this patient would not be benefited with ODC1 inhibitor. Rather, this patient would be. So we need to profile the patients first, and then only we give them the therapy. Similar was the case with other genes, RRM2. This was pretty uniform, like most of them were having upregulation. Same in cases MOX2, SAT1. If there was something that was upregulated in all the patients, yes, that we can say as a master target. If you remember my previous lectures, there were quest for master target. This is what the industry looks for. They're looking for master targets. But in cancer, I can tell you this is impossible because every patient is different. And overall, this was all done in the cell lines. So this was even in cell lines, ODC was showing a very uniform behavior, but in other ones, there were different responses in terms of the expression level of these genes. And we use the Tachman chemistry for validating these genes. So we did genomics, we did transcriptomics, and we did in silico metabolomics. We were not able to do proteomics, and because I left that institute before we could finish that study. But we did combine genomics and transcriptomics for another purpose. We used this fusion of genomics and transcriptomics, and we created signatures for classifying the patients so that we can categorize the patients, and then we can give them the therapy as per their subtypes. This was in collaboration with another scientist, Professor Hautaniemi in Finland, and he's a computer scientist. He's not a biologist. He's not a clinician. He does not know anything about cancer, but he did this study, and we were able to have 3 different classes of patients with 3 different survival rates. And these were the survival statistics for these patients based on the data that we provided to them. So see, when we fuse the transcriptomics data and the genomics data, we can use it even for finding out the group of patients, and we can tell them that how are they going to progress in their disease. So one group of patients with the red line, they have got the poorest survival probability, right? And the blue one is better and the green one is the best. So if a physician has to make some decision about treating them with certain drugs, they can have some information about which one to prioritize and which one to be given an alternative therapies. Then we also use this data for creating some prediction models, support vector machine-based models. I had a student from Africa, and he developed this prediction models to tell about the cancer in normal samples. So he also had, but this was a computational method where we did the feature selection, and then we used the support vector-based, machine-based prediction models. This was the story that we did for finding out the target. Am I audible still?
Yes, sir.
Okay, let me just mute this notification I'm getting. All right, sorry. Sorry about the disturbance. Somebody's trying to call and it's coming on my computer as well. So therapeutic targets can also serve as good biomarkers. So the multiomics exercise that we do, it's not just about finding targets. We can also do it for risk prediction. We can find out biomarkers. We can use it for classification. And the ultimate objective is to help the patients. So therapeutic targets can also serve as good biomarkers. So when we are looking for the genes, or we are looking for the proteins, or we are looking for the targets that are altered in tumor cells, they can also be used to tell us about the progression of the disease. They can also be used to tell us about even for the diagnosis of the disease or for the response to the drugs. So they are known as diagnostic biomarkers, which means anything that we can measure and it can tell us whether there's a disease or not. We call them as diagnostic biomarkers. Then prognostic biomarkers are the one which are the measurable entities that can tell us how the disease will progress. Will it be fast? Will it be slow? Will it be worse? Will it be benign? And then we have got the predictive biomarkers, which can tell us that whether this particular drug will work or not. So these are known as predictive biomarkers. So from the same study, we tried to pick up some of these molecules, some of these targets for serving as biomarkers. And there was another study, before I tell you my story, this was another paper that utilized the same multi-omic data set. And yesterday I saw some interest in some people who are trying to do the meta-analysis. So I thought of putting this slide for them, that there's a lot of scope because there's a lot of data that you can utilize and do these meta-analysis and find out the targets and biomarkers. So they have utilized the TCGA, the cancer genome atlas, and gene expression omnibus. And then they did the statistical analysis, machine learning models. But you have to, if you validate these studies in the in vitro and in vivo conditions, they become very good reports. You can publish them in high quality journals. And then you get the biomarkers, which are diagnostic, prognostic, or therapeutic, based on different omics that they have taken out. So this is just to give you a reference for doing some of the studies that you are interested for multi-omics. Okay, coming back to the same slide that I had shown before, we have got other genes that we utilized for the biomarker analysis. So there was one that is here, MUC4, which was downregulated, and there was another one, which is ADAM12, which was upregulated. So we thought of choosing these two because these are significantly altered at the DNA level as well as the RNA level, and utilize them to see whether they are good biomarkers or not. So same approach, but for a different purpose. So we have utilized it for identifying the targets. We have utilized it for the classification of the patients. Now we are going to utilize it for identifying the biomarkers. Now we want to use it as prognostic biomarkers, which means we want to know how the disease will progress, and if these two molecules can tell us about the different stages of the disease. So for that, we needed a cell line model because you can't do these studies in patients unless you have got a very good data at the preclinical and cell line level. So we choose these three cell lines, CCD841, which represents a normal colon cell, then SW480, which represents a primary colorectal cancer, and SW620, which represents a advanced metastatic colorectal cancer. So we check the expression of these gene MUC4 and ADAM12 in these cell lines to know if there is any changes in these states. If there are any changes, yes, we can use them as biomarkers. So there was another part of the story where we did not utilize the entire gene. Rather, we tried to look at the exon level. So we looked at different transcripts. So what you're seeing here is ADAM12 all, which means it's a complete gene. But then there are different variants, 2, 4, 5, and 12, 13. So these are different transcripts of the same gene. I'm not sure how many of you are familiar with alternative splicing, but with alternative splicing, I touched upon yesterday that with alternative splicing, you get the different formats of the gene from the same pre-mRNA. So we utilized not the gene itself, rather the transcripts, that isoforms we call, for use as biomarkers. And we quantified them in different cell lines, and we tried to find out if there are any changes. Then once we have these targets or we have got these biomarkers, there has to be some preclinical models to test them, right? I've just put this single slide here, but the entire paper is available. I've given the reference here. If you are interested, you can go there. I'm trying to make it very simple as well as useful. And we have a diverse audience. So all that we found here was that yes, Adam 12 and MUC4, both of them can serve as the biomarkers, the prognostic biomarkers for these for the colorectal cancer progression. Now, once we have the targets, the problem, especially for the personalized approach, that how do we test them? So there has to be either we take thousands of mouse, or we take thousands of cell lines, or we take patient-derived organoids, which are 3D structures, or there was somebody very smart, and he was a good friend of mine. Now he's relocated to Ireland, and he made some fly avatars. This is Drosophila. So what he did, he took the patient's genomic analysis. The only problem here was he did not take all DNA, RNA, protein together. Rather, he just looked at the mutations. But then he took these mutations and he made the engineered these Drosophila and made it as good as this patient. And then, since Drosophila you can culture in the petri plates and you can culture many of them, they were able to test thousands of drugs on it. And then with that, they were able to find out which treatment is going to work and which treatment is not going to work. And then they give you 5 treatment options for that patient to be implemented by the oncologist. So this is now already a company that is being run by Laura in UK. And I told you another company, Nantomics, which is also doing multi-omics rather than just the DNA mutation profile. So they had identified different oncogenes and tumor suppressor for different variants. They have done it for a couple of cancers, but the final outcome for all this is to be precision and personalized medicine. And that's what we are hoping to do with the multi-omics as well as the personalized medicine approach, that we can provide the right treatment to the right patient at the right time. I think with that, I will stop here and take any questions if you have. We have got 8 minutes to go. If you want me to go through any slide again, I can do that. And if you've got any questions or discussion points, feel free to let me know. Okay, so I'm hanging around to try to absorb all of this, and I can run the slides once again so that you can have an idea. Or if you're seeing it on YouTube, you can even scroll back and see that. But for you to be able to see it.
Thank you very much, sir, for this highly informative session. We'll wait for a few more minutes for people to ask some questions, and then we
Okay,
will move on to the next session.
all right, that's fine. So I'm turning off the camera, but I'm hanging around. So if you have any question, you can speak or you can write down. Thank you. Thank you for your listening. Well, Sandeepan, these models, they were validated first, that whether they actually recapitulate the human system and human mutations. So validation means first they were tested with the drugs, and then it was also tested in other animal models. And it was found that yes, this was working. So the considerations here are numbers. If you can make as many tests as possible, that is the best. So if you test only 10 drugs and tell, okay, these two will work, it will be inferior to what if you say we have tested 1,000 drugs or 5,000 drugs, and then we are giving this recommendation. I would say increase the number of drugs that you test, and then give the recommendations for which drugs are going to work and which one is not. But that is going to be expensive as well. And that's why this is unaffordable for patients in India, that it's costing about $100,000 for a patient. Okay, DNA changes do not always correlate with RNA level changes. Why is that? Okay, the first and simple answer is epigenetics. So DNA might be duplicated, but there is a silencer of methyl group there. It might not express at all. If you're seeing something duplicated at the DNA level, it would not be transcribed into the mRNA. I hope that answers your question, Mustafa. And there are other mechanisms as well. Swati, I have made Hibzer as a co-host.
Yes, sir.
Okay, good. So in 2 minutes, we'll reconvene.